Go ahead and hit. There we go. Margaret got it. Okay.
Go ahead, Taylor.
Okay. Good afternoon. I'm Taylor Doherty from Allergy Immunology, and it is my distinct pleasure to welcome Dr. John Lyons to give medicine grand rounds today. Dr. Lyons graduated with a BA in chemistry from Pomona College in 2003 and earned his MD from the University of Southern California in 2007. Dr. Lyons completed residency training here at UCSD in internal medicine and completed that in 2010, and then served as a chief resident in internal medicine in 2011. He concluded his formal medical training in 2014 at NIAID as a clinical fellow in allergy and immunology. Following completion of fellowship, John was selected for the NIAID transition program in clinical research, and he served as an assistant clinical investigator in the Laboratory of Allergic Diseases until 2018, when he was selected as a tenure track PI. Since 2018, Dr. Lyons has served as the chief of the Translational Allergic Immunopathology Unit, where his lab seeks to identify mechanisms and pathways critical to allergic inflammation and hypersensitivity that can be targeted in order to conduct first-in-human trials that prevent severe reactions and inflammation. In 2015, Dr. Lyons received the Quadruple AI Foundation Mastocytosis Society Research Award, and Dr. Lyons has also received NIAID Merit Awards in 2016, 2017, and 2020 for his work related to the identification and characterization of hereditary alpha tryptosemia as a common genetic trait and modifier of mast cell associated diseases and reactions. His seminal 2016 first authored publication in Nature Genetics has been cited over 230 times and represents a critical and clinically relevant discovery in our field. Dr. Lyons was named a Lasker Scholar in 2018, and in 2020, he received the Young Physician Scientist Award from the American Society for Clinical Investigation. And in 2021, he was awarded the Innovation in Research Award by the Mast Cell Disease Society. So on a personal note, I have known John since we worked briefly in the lab together, and my claim to fame is that I know John from that time, and I could say we worked in the lab together. This was in 2009 at La Jolla Institute in Mick Croft's lab, and this was during his residency research rotation. We were very sad, of course, not to keep him for allergy fellowship at UCSD, but it has been absolutely amazing to see his development as an outstanding physician scientist at NIH. We are also honored for one of our own IM grads to give grand rounds. Today, Dr. Lyons will present Harnessing the Power of Immunogenetics to Understand and Treat Hypersensitivity Reactions in Humans. Dr. Lyons, it's all yours.
Thanks, buddy. I wish I could be there in person, obviously. At least we have sort of the advent of Zoom that everybody sort of, it's a thorn and a rose, I guess, at the same time, that I can at least do this virtually, and hopefully it won't be too long from now that I could see many of you again in person. I was asked sort of, well, first of all, there's the CME thing that I was asked to put on there. So scan the QR if you guys need to do that for CME. I was asked to give sort of a vignette. And as Taylor mentioned, a big part of the reason why I chose the career path of a physician scientist really studying allergic diseases started with that sort of 2 months in Mick Croft and Bruce Serra's labs under their mentorship, and with Taylor kind of guiding the way, where I really kind of got my first seat at a flow cytometer and began to understand a little bit about science and a little bit about immunology. But sort of shortly after that, I was on the nephrology rotation. I think it's just like a 2-week rotation back then, at least. And Scott Mulaney actually was the attending, if I remember correctly. And we were consulted on this 21-year-old kiddo at Hillcrest. He was an adult, but he looked like a kid. He was quite small and had a lot of chronic problems, including autoimmune kidney disease. I think he'd had at least one failed renal graft at that time. He was, I think, on his second. Whopping peripheral eosinophilia, eosinophilic esophagitis, additional enteropathy, and a bad dilated cardiomyopathy. And he was kind of going into a hepatorenal type syndrome, really. And I think we were really called essentially to UF him. But at the time, the disease smacked of something sort of immunologic, even though at the time I didn't know what it was. So I remember talking with the primary team, and they reached out to consult Allergy to try to figure out if there was something sort of more to this. And at the time, actually, Taylor was the attending on service, and Luann was the fellow. I think she's faculty now at the UCSF, if I remember correctly. And what they found was in this patient, sort of a unifying diagnosis where there was a mutation, damaging pathogenic variant in this gene FOXP3, which leads to a reduced number of regulatory T cells, which are sort of shown here on the right, and is linked to this disorder known as immune dysregulation, polyendocrinopathy, enteropathy, and X-linked syndrome, or IPEX. And that's really what caused this patient's presentation. And it was the fact that, we saw this guy at Hillcrest that kind of struck me, and it made me realize, in medical school, a lot of times we learn about sort of the common things and the severely bad things, right? Like if it's either it's common or it's gonna kill you. And what struck me was the fact that here were these supposedly or truly rare diseases that you could see in severely ill patients. And this kind of began the past decade plus journey I've been on, trying to study more and identify more of these disorders in order to better treat patients, not just like this, but that have common disorders related to that. So here's the crew at the NIH. I would be remiss not to comment on two additional postbacks who aren't pictured here. Jack Shavanik on the bottom right and Jawan Kim sort of more centrally located because of their important role in a lot of what I'm going to talk about today, as well as highlight two great folks in the lab, Yihui, who's my biologist, and Alyssa James, who's a really kind of a rising star clinical fellow who really played an integral role in the sort of the second story I'm going to talk about today. So over the course of the day, I really hope I can emphasize to you all the importance of considering what we might consider zebras, these rare genetic disorders that can lead to really substantially impacting clinical phenotypes, and in mass are really probably more common than we give them credit for. I will talk about one of these disorders that actually really is quite common, this genetic trait that we discovered in the lab called hereditary alpha-tryptosemia. I'll talk about a number of sort of clinical applications and how this trait's really kind of changed our understanding of ways to interpret an important biomarker in allergic and myeloid disorders, but also kind of gives us some additional mechanistic insight into why some people have bad allergic reactions. And then in the second sort of phase of today's talk, I'm going to speak a little bit about a very rare disorder, but really an illustrative one of how we can really kind of personalize treatment approaches to patients with bad allergic inflammation and disease, such that we can really improve their lives when we understand the immunogenetics governing their pathology or the pathology underlying their presentations. And that's really kind of the fourth point to this. So the money is in the genes, at least in my opinion. The way I view allergic diseases, generally we think of these things as complex genetic traits, but to back up a second, why do we even think about genetics when it comes to allergy? And importantly, the number one sort of risk factor for allergic disease is a family history. And so of those patients who present with moderate to severe atopic dermatitis, we're talking about almost 3/4 of their parents have a family history of allergy. If you have a parent who is allergic, at least one parent, you're 2 to 3-fold have a 2 to 3-fold higher risk of developing allergic disease. And at the population level, 2 to 3, odds ratio of 2 to 3 is actually quite substantial for those of you who don't think about sort of like population level genetics. And finally, if both parents are atopic, the risk increases to 3 to 5-fold. And moreover, if you're a monozygotic twin, that even multiplies 2 to 7 times that initial risk of 2 to 3 higher with one parent. So you can see that this is sort of a compounding risk, and it really suggests a strong genetic driver for allergic diseases in general. A lot of people sort of say, well, that's fine, but it's complex genetics. It's a lot of different variants and a lot of different genes that sort of all together add up to cause a problem. And that, that's likely and well true for, sort of garden variety allergic rhinitis and some of those kinds of things. But when people have looked at this a little bit more carefully, and this is certainly most well studied in the pediatric population, what has been found and reported is that when we think of rare variants as sort of these rare single gene disorders sort of en masse, like all together, it's 1 in 100 live births is estimated the prevalence of someone being born with a single gene disorder. And importantly, among those who are sick, right? So like most kids don't end up in hospitals, but if we talk about sick kids, at least in Ohio at Rainbow Babies and Children's Hospital, it was determined that nearly 3/4 of admissions were associated with single gene disorders, leading to the cause for the admission. And finally, there was this great paper out of Baylor that Jennifer Posey kind of led the effort on in Jim Lupsky's lab that demonstrated not only are these single gene disorders fairly common, but that many people who present with complex syndromes actually have multiple single gene disorder diagnoses. So this is showing you that approximately 5% of the patients that they identified with a single gene disorder had two. This would be like Kabuki and Tay-Sachs or things like that, where there's multiple identified known single gene disorders that are leading to these clinical presentations. What about the immune system? Because a lot of these, particularly historical clinical genetics is related to metabolism and glycosylation disorders and things like that, some of which have immunologic consequences, many of which do not have at least direct immunologic consequences. But with the advent of next generation sequencing and the solving of the human genome, we're talking 2 decades on now, the number of single gene disorders leading to immune problems has skyrocketed such that now, this is data from 2022, so we're probably well over 500 now, known single gene disorders leading to inborn errors of immunity or IEIs. And importantly, of these, it's estimated that at least 10% present with severe allergic phenotypes as a primary presenting feature. And we coined the term primary atopic disorders for this sort of subset of these IEIs where severe allergy is really a primary driver. And it's really been this group of disorders that I've been highly focused on for the last decade plus of study. And so our approach has been fairly simple and one that's, I think, been really championed at the NIH, but certainly isn't limited to there and certainly can be applied really anywhere. And that's really of a bedside to bench and back to bedside approach, where we identify a patient who is severely inflamed, highly atopic, or having bad kinds of reactions. And we seek to identify the genetics and mechanistic underpinnings of that clinical phenotype, such that we can identify specific pathways that we can sort of co-opt or create new treatment strategies in order to improve the lives of our patients. So this sort of translational bedside to bench to back to bedside approach. And certainly this is not unidirectional. And when we look at a lot of studies such as some of the early anti-IL-5 studies in patients with asthma, the response to therapy actually informed the genetics and similarly, using some gene first approaches, as I'll kind of demonstrate later, we can similarly inform patient phenotypes and identify specific or unexpected clinical presentations associated with known genetic pathologic variants. So in that vein, I'm going to talk about 2 stories today. One that I think is really illustrative of how we can kind of harness this approach to identify even common genetic variants that have sort of broad impacts on allergic and myeloid processes. And then the second is this rare disorder that I'll discuss, where we can really sort of demonstrate the power of using this kind of approach to identify specific and directed therapies that are really efficacious in our patients. So hereditary alpha-tryptocene. So this is a genetic trait that we identified, first reported back in 2014. But to back up a little bit, what is tryptase and what's that all about? So tryptase is a neutral serine protease. It's almost exclusively expressed by the tissue-resident effector cells of allergy called mast cells. These are myeloid lineage cells that are born in the bone marrow and get recruited to sites of inflammation, but also just get recruited to mucosal and cutaneous surfaces. So they line the gut, they line the lungs, they're in the skin, they're in inflamed joints. There are probably some in non-inflamed joints as well. And there's some that reside in the bone marrow for reasons we don't fully understand. And it had been known for quite some time that this serine protease, this tryptase that's expressed almost exclusively by these mast cells, is elevated or higher, sort of like a bimodal type distribution, as you can see here, in about 5% of the general population. And it wasn't really appreciated why that was the case. And there are other immunologic sort of molecules and markers that do similar things, like even immunoglobulin IgG has a similar distribution that may be also dictated by some genetic cause that's not yet determined. But back in 2014, we realized that at least at the NIH, we had 4 families that I was seeing as a first-year fellow that were being studied by a couple of different investigators that were inheriting this high tryptase in an autosomal dominant fashion. And it was associated with a number of clinical features, including symptoms of mast cell mediator release and anaphylaxis. And as we've studied this more and more, and others have as well, what we found is that basically everybody with few exceptions, who has a high tryptase has it because of this genetic trait called hereditary alpha tryptosemia, which affects about 5 to 6% of the general Western populations in which it's been studied. Now, importantly, this biomarker had been associated with a number of disorders, including clonal and non-clonal mast cell disorders, myeloproliferative diseases, where disorders like CMML, AML, MDS, and polycythemia have been associated with high serum tryptase levels. And importantly, there's a rich literature sort of historically on severe anaphylaxis to stinging insects, most commonly Hymenoptera species. So we're talking about wasps, hornets, yellow jackets, that is associated with having a high baseline serum tryptase level. And so now that we understood why most people have a high tryptase, we had to sort of re-examine a number of these phenotypes in order to get a better understanding as to what was what and how this was playing a role in these different disorders. To back up a little further, the genetics of tryptases are fairly complicated, so I want to spend just a minute on that. And importantly, these genes are very recently evolved in humans. In fact, orthologs in mice and other animals are not good surrogates for these genes, making them really almost exclusively human, and probably also explains some of the racial and ethnic differences we see in different tryptase gene composition that I don't really have time to get into today. But importantly, there are at least 4 known human genes that encode tryptases. There's gamma, which is TPSG1, beta, which is B2, alpha beta 1, which is AB1, and delta, which is D1. Delta has a large chunk deletion, so we mostly ignore that, and gamma has a transmembrane domain, so it's not secreted. So at least for the purposes of this talk, we're really talking about TPSB2 and TPSAB1 when we talk about tryptase. Importantly, we know that TPSB2 almost exclusively, with one rare exception, is only encoding of different isoforms of beta tryptase, whereas TPSAB1 can either encode alpha or beta tryptase. And these genes, as some of you may see here at 36 kb, are really tightly linked together, such that if you guys remember sort of your basic genetics, these genes are in linkage disequilibrium, meaning that you inherit them together from a parent as a haplotype. And based upon that, for a long time, we thought that there were basically 3 genotypes resulting from the 2 possible haplotypes that you can inherit. So you get 2 betas from mom and dad both, so you have 4 beta, and that's about a third of the population, 30%. You get 1 alpha on one allele or one haplotype from one parent, or 1 alpha on the haplotypes from both parents. Remember, you're only going to get 1 copy of genes from mom and 1 copy from dad. And these are the 3 sort of canonical genotypes that we knew sort of going into all of this back in 2014. But importantly, those of you that are quick at math, you'll realize that we didn't add up to 100% right there, because the remaining individuals have this. So they have extra copies of TPSAB1 encoding alpha tryptase at that locus. And that's really the defining feature of hereditary alpha tryptosemia. What was a little bit strange to us is that the average tryptase level, or really the median tryptase level in the general population, is about 4. But if you have hereditary alpha tryptosemia, just 1 extra copy, all of a sudden your average level or your median level really is about 13.5, which isn't really explained by copy number, because if you had 1 extra copy and you started with 4, you should really have 5. But it turns out by cloning the locus and long-read sequencing and assembling it in silico, what we were able to discover is that the replication, the alpha duplication, or alpha dupe as it's shown here, is actually a natural overexpression model. This duplicated copy is in essentially every individual we can identify is overexpressed due to co-inheritance of this overactive promoter element that's upstream of its transcriptional start site. And based upon this knowledge, we were actually able to remodel what a baseline serum tryptase level should be in the general population. And there's 2 important impacts of this kind of approach. The first is that using a serum tryptase is a minor diagnostic criteria for the clonal disorder systemic mastocytosis, which is a relatively normally indolent neoplasm of the mast cell compartment, driven most often by a gain of function in the stem cell factor receptor KIT. Importantly, what we were able to do is basically, in this case, model the serum tryptase level based upon the replication number, since we know that each replication basically gives you this stepwise increase. And what I hope you can appreciate here on this log-log transformed graph of the number of replications on the x-axis and the baseline serum tryptase level on the y-axis is how linear this relationship really is, with an excellent fit. And obviously, it's not a slope of 0. And importantly, based on this modeling, we were able to devise new clinical reference ranges that we proposed and published, where based on the number of replication number, provided that it's encoding alpha tryptase, we can determine, sorry, do I need to do something with closed captioning? I'm sorry, just seeing this.
You don't have to do anything.
Okay.
You can just ignore that.
Okay, I just didn't know if people needed to be able to read if and couldn't hear something. So okay, so based on the number of replication numbers, you can see that we were able to identify not only what should be the average number, but also an upper limit of normal. And this, to our knowledge, this is really the first time that genetic understanding of a biomarker has been used to define, really basically define a personalized clinical laboratory reference range. And so it's sort of a new way of thinking about personalized medicine, where in the future, it may not just be tryptase, but a number of these other biomarkers, where PSA is one that's been proposed, where there are genetic determinants that really define what's normal for any given individual. And this has important implications, both for the diagnosis of mastocytosis, as I mentioned, but also for sort of an indication to consider further workup or even treatment approaches to patients with suspected clonal myeloid or mast cell diseases. And I'll kind of give you a real world example here now. So we, based on this, went back and looked to see of those patients who we've evaluated or our colleagues have evaluated, where we have bone marrow data and other data to really know what the clinical phenotype is, and that we've genotyped, how often do we see a high tryptase in the absence of either this systemic mastocytosis or hereditary alpha tryptosemia? And in looking at 670 patients, what we found is that there were only 12 with a tryptase above 8. And of those, our cutoff would be 11.4 to be considered normal. We only found 5. So it was less than 1% of the 670 that we screened that we could find who didn't have one of these other known diagnoses and had a high tryptase without HAT as the cause or systemic mastocytosis as the cause. We deep sequenced bone marrow aspirates from these patients, actually both the granulocytic and the monocytic or mononuclear fragments. And what we found is a number of kind of smoking gun type variants, I'd like to call them, within that were somatic in origin, including a TET2 and Notch2 nonsense variants in 2 different patients, which really suggested an evolving clonal myeloid neoplasm of some sort that didn't really meet any current diagnostic criteria, but could be considered something akin to clonal hematopoiesis of indeterminate potential, or CHIP. Taking a step back from sort of the more recent genetics, when we first identified the patients with HAT, there were a number of clinical phenotypes, as I mentioned, including anaphylaxis and symptoms suggestive of mast cell mediator release that were linked to this genetic trait. And we wanted to see whether this was a valid association. And so this approach has been actually cited a number of times as kind of a unique and interesting way to do this, and one that's gaining sort of traction amongst clinical geneticists. And this is sort of the genotyping first approach. And so in order to try to validate some of the symptoms that we saw linked to our patients, what we did is we reached out to a collaborator in Genome, a gentleman by the name of Les Biesecker, who's got this fantastic cohort of patients called the ClinSeq cohort, where these are healthy volunteers that have donated time and information and tissue and blood samples, as well as consented to not just genotyping, but exome and genome sequencing, where it's already actually been performed, that you can then go and look for your variants of interest and then ask the question, what clinical phenotypes might be associated with these variants? And so we recruited 100 patients from this cohort. We successfully actually enrolled 91 of them, co-enrolled 91 of them, and looked to see what symptoms kind of panned out, what actually looked like it was going to actually be linked to hereditary alpha tryptosemia. And you can see that there are a number there that were significantly associated, although with a small sample size, we obviously couldn't control for multiple comparisons. But the most impactful, the most interesting to us was the severe venom reaction, given the astronomical odds ratios and relative risks, which are likely overestimates based on small sample size. That has been published a number of times. That tends to happen. But we still thought this was a strong enough signal that we sought to go after it. And so we teamed up with a group from Slovenia, which is really a fabulous group led by Peter Karashek there. And what they have the advantage of is they have a nationalized health system and a single center for the entire country, where every patient with a sting reaction, whether it's mild or severe, all goes for evaluation of stinging insect allergy. And so what we did, well, Jack did, my postback at the time, was to genotype all these samples and based on, and then look back at the baseline serum tryptase level, the genotypes, and the severity of anaphylaxis. And here I'm showing you the baseline serum tryptase level and anaphylaxis grade, with 1 being sort of a diffuse cutaneous type response, and grade 4 being really usually syncopal hypotensive syncope. And what you can see, as has been published in the past, is that there's this sort of modest increase in baseline serum tryptase level among those severe reactors, those grade 4 reactors, relative to the rest of the group. What hadn't been discovered or reported before is that this increase in baseline tryptase level, to a large degree, not entirely, but a lot of it is driven really by hereditary alpha-tryptopenia, shown here in red. And we went on to look at this in a prospective manner and saw basically the exact same thing. And when you add sort of the results of the 2 studies together, what you find is that the relative risk for severe anaphylaxis among those with venom allergy is roughly odds ratio of roughly 2. So you're about twice as likely to have a bad reaction if you have both. Importantly, in both studies, the prevalence of HAT overall was no different than the general population. So this is not that HAT leads or is associated with a higher risk of developing an allergy of sorts. It's just that if you're have bad enough luck to have both, you're about twice as likely to have a bad reaction. The other thing we noticed in this initial cohort is that a number of patients, in fact, all of the patients we found who had HAT and systemic mastocytosis, were in that grade 4 reaction group. And more importantly, the prevalence of systemic mastocytosis among those with HAT was much higher than we had anticipated seeing, suggesting that there might have been association. And as I mentioned sort of at the outset, high tryptase is linked to systemic mastocytosis. So we were interested in sort of ferreting this out anyway. And so, at the time, we were the first to look. Now, 3 additional studies have come out, one from the UK largely, one from the EU that's largely based in Austria, Germany, and Poland. A large cohort, the largest actually, is from Italy, the Sordi et al. Cohort. And then there's a much smaller study that was performed in Michigan, all of which found that among patients with systemic mastocytosis, there's almost a 3-fold increase in the prevalence of HAT relative to the general population, which, as I mentioned at the outset, was about 5 to 6%. Importantly, we and others have seen that among those with systemic mastocytosis, in a manner quite similar to what we see with Hymenoptera venom allergy, is that the patients who have both, sort of shown here on the far right, where it's 2, 3, 3, 2, and 4, 2, are much more likely to have a history of severe anaphylaxis. And indeed, in the EU study, they demonstrated that the patient's symptomatology, as it relates to mast cell mediators and things using a validated symptom score, was more severe and had a gene dosage sort of relationship with higher numbers of alpha tryptase copy number. Beyond systemic mastocytosis, it's not just that disorder, but we looked in patients with idiopathic anaphylaxis and found effectively the same thing, where the prevalence was about 3-fold as high, again suggesting that whatever causes idiopathic anaphylaxis, which we still don't know, is modified or made more severe by also having hereditary alpha-tryptosemia, such that you're more likely to actually present with anaphylaxis as opposed to milder symptoms. And in this ongoing study in collaboration with Laurie Children's, we see a similar situation in food allergy. And this is just sort of new data, kind of hot off the press, but these are kids with a history suggestive of peanut allergy who underwent challenge to peanut and reacted. And what you can see is that with increasing amounts, see there's 2 different scoring systems on the left and on the right. And that's the scoring systems are shown on the y-axis. And you can see moving from left to right in both graphs, an increasing amount of alpha relative to beta tryptase, with those HAT folks being on the far right. And you see quite nicely this sort of gene dosage impact on the severity of reactions, even despite the small sample size and number. And so, taken together, it really suggests that having HAT, at least, and certainly having increasing amounts of alpha look to be associated with having more severe reactions. So we wondered how in the world that would be the case, because alpha tryptase doesn't have any enzymatic activity. And so here I'm showing you a schematic of tryptase generation in a mast cell, where these proteases, like a lot of other proteases, are initially generated as zymogen and require proteolytic activation in order to become these mature tetrameric toroidal shaped proteases, where the active sites really reside in the middle of that donut. These are stabilized by heparin under a low pH, and I'm showing beta here because at least sort of the historic literature would indicate that beta is the only enzymatically active protein. And so we were sort of stuck with the quandary, really. Well, how is it that overexpression of alpha tryptase can lead to anything if it's a dead protein? It turns out that we don't just make homotetramers, but that naturally heterotetrameric tryptases are naturally occurring in mast cells and have some unique properties. And we think these unique properties are imparted by the fact that alpha tryptase, at least as a homotetramer, doesn't require heparin to remain stable even at neutral pH. And so we believe that these heterotetramers have, despite having loss of enzymatic active sites on the alpha subunits, are actually more stable and gives them longer dwell time, such that they're able to cleave specific targets at lower KDs, such that they can have clinical, we would think clinical, certainly in vitro activity that beta homotetramers do not at the same concentration. So one of these targets is EMR2, which is a adhesion GPCR that has this really interesting non-covalently bound extracellular fragment that's tethered to the extracellular space via glycosaminoglycans, such as dermatan sulfate or things like that. And it turns out that like shearing forces can actually drive dissociation of that non-covalently bound fragment and actually stimulate a signal that can increase a mast cell's reactivity to a vibratory stimulus. And perhaps the most sort of extreme or interesting example of this was a paper from D. Metcalf's group at the NIH back in 2016 of a family, actually a couple of families with the same variant in this gene, or at least the ADGRE2, which encodes EMR2. And what these patients had was severe vibratory urticaria. And so what Hirsch Kamaro did from Dean's group is he actually took the patients, put their arm on a vortex to see what would happen in their skin. And indeed they hived, but impressively what you can see here is that on the left is without stim, on the right's with stim or vibration, and all of the red is stained for tryptase. And so you can see this massive release of tryptase into the skin of these patients upon vibration, indicative of mast cell reactivity and activation via disruption of this EMR2. So we asked the question, what about in just healthy folks? Like how does this heterotetramer impact potential reactions to vibratory stimuli? And it turns out a good number of people actually respond or react and hive up when you put their arm on a vortex for a minute and a half. But what you can see here is that based on sort of the severity of the response, which is indicated by this score on the y-axis, is that as you increase the amount of relative expression or germline really, composition of alpha to beta tryptase, that you see more and more severe reactions among those with higher and higher ratios. And this didn't matter whether it's hereditary alpha tryptosemia or just people with increasing amounts of alpha, really suggesting that HAT is the sort of the tail end of this continuum and sort of an extreme outlier that helped us identify this phenomenon that occurs in basically everybody. In my lab, we were also able to show that PAR2, protease activated receptor 2, on endothelial cells is a specific target of heterotetramers. And so this is using a transwell system. So you basically have a monolayer of umbilical vein endothelial cells sitting on a membrane with FITC dextran on the top. And then you just measure the amount that passes through the membrane and through the cells over time, depending on how you stimulate them. And you can see that the cells are a little leaky no matter what. So that's shown here in black down at the bottom, untreated. When you hit the cells with either histamine or heterotetramer, you can see in green or teal and red that you get a nice permeabilization, so a nice increase in the amount that goes through that membrane over time. And that if you take peptide antagonist of PAR2 or beta homotetramers, you can see in these dashed and dashed lines that you really have no significant difference compared to the untreated condition. And this leads to this paradigm of a way in which, at least in vitro, that heterotetramers can promote vascular leak from human endothelial cells and may potentiate severe allergic reactions that are characterized by hypertensive syncope in particular and vascular leak. You know, I mentioned at the outset that not just mast cell disorders, but myeloid neoplasias are also associated with a high tryptase often. And so that got us sort of interested in trying to determine what impact HAT might have in particular in readily available data. And so I indicated, but I don't know that I explicitly stated, the replicated TPSAB1 encodes wild type or canonical alpha tryptase. So there's no different in the coding sequences between the replications and the endogenous loci. But what there is a difference in non-coding variants that can tag duplicated and distinguish it from wild type. And so we can go back into RNA sequencing data sets and look at the prevalence of HAT among those expressing tryptase, right? We can't see this in, if there's no tryptase RNA, we can't say anything. But among those expressing, we can compare those with beta versus alpha versus duplicated. And we did that. And so we looked at, in collaboration with Chris Horrigan from NHLBI, we looked at the Beat AML data set, which is a really nice cohort of patients that was published not too long ago in Nature, where they have linked RNA sequencing, exome sequencing, and metadata, immunophenotypic data, including survival. And so what we looked at is just, we asked the question, does HAT impact survival? And importantly, among systemic mastocytosis, there's a hint that you have a more indolent course and less aggressive features, at least histologically, among patients who have HAT and systemic mastocytosis. And so when we looked at patients who didn't express tryptase, and we had data from exome, whether they were expressing alpha or not, we really didn't see any difference between those that expressed alpha and those that didn't express alpha when their blasts weren't making tryptase. And I should emphasize the fact that among AML patients, it's the myeloblasts that are actually the source of high tryptase in these patients. However, the story changed when we looked at those expressing tryptase and which tryptases they were expressing. So when we looked at patients who were only expressing beta tryptase, we saw actually that they portended a slightly less favorable survival, whereas among those expressing some amount of tryptase, but not replicated tryptase or not HAT, we saw a slight improvement. And interestingly, among the very small number that we found who had HAT based on RNA sequencing data, we saw even a further improvement in survival. And so this is really preliminary data, but we're trying to understand how and why this might occur. And one hint might be in the gene expression profiles of these myeloblasts. So this is looking at 4 different data sets, including the Beat AML cohort, that are subcategorized by whether they either were expressing or not expressing tryptase, expressing wild type versus beta, or expressing duplicated transcript versus beta. And what you can see among this cluster up here, TPSD1, TPSB2, AB1, and CACNA1H, that is all the tryptase locus. CDK15 and MS4A2 are also highly enriched in mast cell lineage cells. But the one we focused in on was HPGDS, which is a prostaglandin D2 synthetase. And interestingly, there's a story about PGD2 and its impact on clonal expansion proliferation of neoplastic cells. And interestingly, we and others had noted that among at least two different lines, mast cell leukemic lines, where one of them in the dash line here expresses alpha tryptase, that's the LAD2 line, whereas the 4 beta line up here is HMC1 cells that don't express any alpha. You can see that the HMC1 cells much more rapidly proliferate relative to LAD2 cells, which are quite indolent. And interestingly, as we spiked in additional amounts of PGD2, which is really not generated at all by HMCs, but is generated in relative abundance by LAD2 cells, we saw that we could slow down the rate of proliferations quite substantially. And it would suggest that, how it is exactly that tryptases are driving this sort of mast cell-like phenotype among myeloblasts isn't so clear, but having a more mast cell-like phenotype certainly seems to be associated with a more indolent progression or course of disease, and certainly of behavior of the blasts. So we're actually working right now to try to expand this into patients. These data and this genetic trait we have a collaborative research and development agreement with a startup to actually who has a candidate neutralizing antibody against tryptase. But in the time I have left, I'm excited to kind of share that rare story I mentioned, where I think it kind of shows the full power of this approach, where we can identify a patient who's quite sick, identify a cellular phenotype that we can then target with a specific therapeutic to provide really a lot of benefit to our patient. So this was a kiddo that was sent to us from Vanderbilt several years ago now, who, at least in the pathology report, said, this looks like severe allergic contact dermatitis, which I think most of you, seeing this kid would say that. I don't think that's what he has. But he had these severe eczematous plaques. They were chock full of eosinophils. And you can see here, they were quite psoriasiform, but they were really eczematous and inflamed. And it's a little difficult to see in this image, but I kind of highlight here a number of eosinophils that have infiltrated the tissue of one of these lesions. And it wasn't just his skin that was full of eosinophils. His gut was full of eosinophils. He had eosinophils in his liver and his kidneys, and his bone marrow was chock full of eosinophils. But importantly, they didn't look dysplastic. Unlike hypereosinophilic syndromes, they all looked like they were maturing normally. There were just way too many of them. I think his peripheral blood eosinophil count when we first met him was about 12,000. So we undertook exome sequencing at the time. We obviously, most people are doing genome now, but back then, this was a few years ago, we were still doing exome. And we found 3 variants, pathogenic variants in 2 genes that were required for this individual's presentation. And this is a true digenic disorder that we've sort of uncovered in this child. So the first of the 2 lesions was this missense in the fibronectin-like domain of the gene oncostatin M receptor. So OSMR is part of the GP130 superfamily, so this IL-6 superfamily of receptors. This receptor is effectively limited to skin, nerves, other non-hematologic or hematopoietic lineages, with the exception of probably some granulocytes. We have some preliminary evidence. You can see here that the missense was expressed. You can see on the left, we're looking at dermal fibroblasts, Western blotting of dermal fibroblasts from our patient, looking at OSMR expression, and in 2 different lines that we derived from him, and 2 different controls. You can see that the OSMR levels are no different in our patient relative to controls. I should point out that the patient was homozygous, and each parent was heterozygous, but there was no evidence of consanguinity in the family. It's just that the variant, as you can see, is not terribly uncommon with the MAF of about 0.002. So it was just kind of bad luck. The second lesion we identified was a truncation of TRAF6. And so for those of you who are kind of looped in at all to immunology, this is a pretty important molecule in a number of pathways, and particularly propagating NF-kappa B signaling downstream, BCR, TCR, LPS, TLR, other TLRs, IL-17. So it's quite important and present in a number of places. It's a ubiquitously expressed protein. And importantly, the truncation was expressed. So we had this non-sensor stop prior to the last, it was actually present in the last exon, which is actually quite large, and encodes the 2 final domains, including the coil, and TRAF domains of the protein. You can see in cDNA that there was no sort of preference of one being expressed over the other. And on Western blotting of the N-terminus, you can see that we indeed see truncated TRAF6, or this lower molecular weight band, as well as the full-length protein in our patients. Again, this was in dermal fibroblasts. So what are the predicted consequences of these variants? So TRAF6, as I mentioned, is downstream of a lot of important places. But it gets recruited by that TRAF domain. And importantly, to propagate NF-kappa B and MAP kinase signaling, that coil domain is critical for K63 ubiquitination. And so lacking those 2 domains, we would be left with this ring-like zinc finger domain of this truncated protein, which really, to our sort of best abilities, seems to be effectively a dead protein. It may have a decoy effect on some phosphatases, but ultimately seems to be just haploinsufficiency. In terms of the OSMR mutant, it's predicted to be damaging in part because these fibronectin-like domains are critically important for dimerization of these heterodimers throughout the superfamily, but in particular in response to OSM signaling, OSMR and GP130, where the ligation of those 2 drives dimerization, and fibronectin is critical for linkage of those 2 and propagation of JAK-STAT signaling in particular. And so this was predicted to sort of limit that ability to cross-link and signal. So indeed, in primary dermal fibroblasts of our patient, you can see that basically there's no STAT5 phosphorylation in response to oncostatin M. And similarly, there's a substantial reduction in ERK phosphorylation in response to LPS, which really is the primary pathway that's dependent on TRAF6 following stimulation with LPS, whereas NF-kappa B signaling is not as dependent exclusively on TRAF6, and we see less of a decrement. Importantly, despite sort of this intrinsic pathway problem, what we saw is that there's a hyperactivation of both MAP kinase, specifically ERK1/2 and phospho-STAT5 in response to a number of ligands. Here I'm showing you LIF, but it's also in response to growth hormone, IL-6, and a number of other superfamily molecules. And in particular, that hyperactivation of ERK seems to be really kind of a critical problem in regard to some of his phenotypes, including his short stature. And we were able to recapitulate that when we silenced TRAF6 in our control line, which had the same homozygous loss of function OSMR variants. And we're able to similarly sort of recapitulate that hyperactivation of ERK in response to all of those different ligands. Importantly, using a number of different JAK inhibitors, we ultimately identified ruxolitinib, where inhibition of JAK1 and 2 was sufficient to normalize both STAT5 and ERK1/2 hyperactivation in our patients' primary dermal fibroblasts. And we were able to move that into the patient following a long conversation with FDA. So at baseline, prior to sort of really any, well, really these are washed out cells, we can see nice activation of STAT5 in response to IL-7 or IL-15. This is just sort of a surrogate marker of utility or sort of efficacy of drug. You can see what he came to us initially on methotrexate. And actually there's a long old literature of how methotrexate is a moderate JAK-STAT inhibitor, which actually makes some sense as to why it works in ways that I wouldn't have otherwise thought it would until now. And importantly, ruxolitinib, at relatively low dose twice a day, was substantially improved or substantially efficacious at blocking STAT5 phosphorylation. So to kind of give you a sense of what this looked like, this was our patient, showed up to us when we first met him. This is 4 weeks after starting him on ruxolitinib. And you can see, we saw just all of his lesions melt away. And I think one of the most gratifying things was when his dad said, or his teacher told his dad that he doesn't cry when he washes his hand in class anymore. So we've been really pleased with his improvement on drug. So, this is just a couple of examples. Obviously, I'd love to share more, but we've been really interested in a glycosylation disorder caused by PGM3 deficiency, where we identified impairment of N-glycosylation, specifically the complexity of that, and where I'm the PI on an interventional protocol where we gave these patients supplementation to try to improve their symptoms. And we're still trying to understand all of the mechanistic underpinnings of the results that we saw in that study. We described a disorder caused by urban haploinsufficiency, where it was akin to really a hyper IgE syndrome that looked a lot like patients with autosomal dominant hyper IgE syndrome caused by dominant negative STAT3 mutations. And indeed, in both disorders, we found that heightened IL-4 receptor alpha expression was present on naive lymphocytes. And we're able to target that with dupilumab, in particular in collaboration with Cincinnati in that index with urban, who'd really failed to respond to really any prior intervention, including mepolizumab and other biologics, where dupilumab really worked wonders for him. And we're still looking at a number of patients with Jobs who are doing really well on Dupi and trying to understand sort of additional sort of mechanistic insights that we can glean there. And finally, as we kind of look forward, as some of these stories kind of reach more maturity, we have a sort of a nice pipeline of patients that we've identified, including STAT6 gain of function, which should be coming out, which is a big international cohort study that should be coming out in JX Med here in the next couple of weeks. But also patients with both gain and loss of function on STAT9 receptor, germline gain of function in JAK3, and STAT5B germline haploinsufficiency. Obviously, this kind of work takes a large community, and I don't want to take up too much question time to name all of the collaborators and investigators that have been critical to the development of these stories, but I'd be remiss not to mention the Quad AA Foundation, the Mastocytosis Society, Jeffrey Modell Foundation, and Lasker Foundation for their support, in addition to DIR support of the research. And with that, I'm happy to take any questions folks might have.
Wonderful. Thanks so much, John. That was outstanding. And I think through that, your two stories that you presented, you really showed that this methodology can really be applied to detect modifying risk genes, as well as those in your second story that really have a huge impact related to treatment. So very wonderful. And I talk to you a fair amount, but sometimes I forget all the work that you've done. So congratulations on all of that. Does anybody have any questions? I think you can put those in the chat. Let's see. So I will start with a scientific and then a clinical question for you, John. I wasn't familiar with the PGD2 story with alpha tryptophanemia or just alpha tryptase inducing PGD2. And as you know, as well as anybody, PGD2 is such a critical mediator of multiple immune and vascular and airway effects during anaphylaxis and severe allergic reactions. Is that thought to also work, because I'm trying to put this together in the pathway, is that thought to work through EMR2 as well that you previously showed data, or how is that happening?
We're not totally sure. So we actually did the ATAC experiment because we thought there's a story by which tryptases are claimed to modify histone and can drive epigenetically drive gene expression. And so we, did that experiment where we paired ATAC-seq with RNA-seq to look to see whether there were different loci being opened or closed. And the experiment was fairly straightforward. We took LAD2 cells that express both alpha and beta tryptases, silenced them with gene silenced them with lentiviral particles, and then sequenced them to see whether we saw differences in accessibility linked to changes in gene expression. And what we saw was changes in gene expression, but no linked changes in accessibility. And so, the possibilities remain that somehow intracellular mature tryptase, enzymatically active tryptase, is still functioning in some way where it's altering or chewing up or somehow modifying the gene expression program without altering sort of the epigenetic landscape. I think that's probably a less likely explanation, but it's a possible one. I think the more likely thing is that there's an autocrine or paracrine effect, like you're suggesting, of tryptases, and the heterotetramers, even though there's less of it, there's not a whole lot of mature tryptase that makes it out of a cell, but some does. And so the heterotetramers might be sufficiently capable of firing through one of these, either EMR2 or PAR2 or one of these type autocrine or paracrine receptors that maybe fly below the radar for beta homotetramers.
Great, yeah, so always more to do, right? So the clinical question I have is that we screen for tryptase levels before, with patients with venom reactions, and you showed very nice data that, there are differences with the severe reactions compared to the lower grade anaphylaxis reactions. But how much of that can then be applied to, or is it being looked at for things like allergen immunotherapy anaphylaxis, or you showed idiopathic anaphylaxis, but are we going to start screening patients almost regardless whether they're at risk for anaphylactic event or they've previously had anaphylaxis with tryptase levels and potentially obviously copy number, et cetera?
Yeah, I mean, certainly first, there's no role for screening asymptomatic individuals. So that's like,
right.
That's park that. The question, yeah, so we get into this sort of gray area, like if you have an allergy and you're going to get shots, right? Like regardless of what it is, should you get screened? Are you more likely to have a bad reaction? I think we kind of need to do those prospective studies, and those aren't terribly hard to do to find out. Like you just need enough people who agree to have a genotype. And then we would have an answer as to frequency and severity of adverse reactions to shots. In the venom story, there was a small signal, but it wasn't a significant one, where it seemed like you might be a little bit more likely to have a bad venom shot reaction, as opposed to the story where you're much more likely to have, actually, if you get a field sting, you're much more likely to have a bad reaction. So I think the verdict's out still. I think there are those who are advocating, particularly the Slovenians are really on this, and they're like, genotype everybody. And then there are others around, Michigan or places like that where they're, don't genotype anybody. So it's like, I think there's going to be a happy medium at some point where I think we will say, particularly in people who've had anaphylaxis before, which is kind of really what we're talking about with venom, right? There it might actually be helpful to risk stratify. And certainly among systemic mastocytosis patients, I think almost everybody's saying we should be genotyping them now, just because the likelihood of a severe reaction and a lot of bad symptoms is much higher, particularly if they also have an IgE-mediated allergy. That's Peter Valent's horse right now. He's saying if you have IgE-mediated allergy and masto, you absolutely have to be genotyped for HAT.
Great. So we'll only see that, hopefully, the commercial labs, et cetera, catch up and be able to get that testing a little bit easier in the future. But yeah, wonderful story. And Dr. Insel had a question here. There's growing evidence of mast cells and perhaps tryptase involvement in solid cancer. do any of the patients with increased tryptase have an increased solid tumor probably risk, I'm assuming, or increased solid tumors?
So, I guess there's two ways to answer that. First, there isn't a literature on having a high tryptase with solid tumors. Really, the literature, at least that I've seen and I'm aware of, really lie within the myeloid neoplasms, probably because in those settings, they have enough of the machinery to make tryptase. Those loci are close enough to open that they can get there when they transform. Certainly, the microenvironment and mast cells within it is an important area of study in a lot of inflammatory and oncologic sort of disorders. There's a lot of people have looked at and tried to understand the role of mast cells in those contexts. I guess the one thing I would say is that baseline serum tryptase levels should be distinguished from tetrameric composition, because baseline serum tryptase levels are really derived from overexpression, really at the gene expression level, of protryptases and their subsequent constitutive secretion as inactive monomers, as zymogens, into peripheral blood. That's what we measure when people have high tryptase in basically all settings, unless you're having anaphylaxis. So unless you're having an acute allergic reaction, you're measuring zymogen, you're not measuring mature enzyme. That said, nobody that I'm aware of has really looked at the impact of differential tryptase gene expression in situ in particular, but even at the germline level of what the gene composition is in people among solid cancers and the role that they might have that's differential, that might impact the behavior of those cancers.
Great. So more to do on that front too, potentially. And that also is the lesson that you can't just use that tryptase level to understand at all what form it's in, the tetramers, et cetera. So I had missed a question by Arnie Gass, fascinating grand rounds. How much does it cost to do this work in an individual patient or at NIH? And then he subsequently says he means in the clinical workup of these patients.
So like tryptase genotyping, is that the?
I believe that, Arnie, if you wanted to add to that, but I believe that that's most likely what you mean, right? Is the tryptase, yes, not just looking at the tryptase level, but then obviously doing the copy number, et cetera.
So we still do it on a research basis, and we're not CLIA certified, so we don't bill, but we're limited in our capacity a little bit. So the company that's doing this clinically currently, at least still the only one right now, is a company called Gene by Gene in Houston, Texas. They run our assay. I believe it's around $150 or so. I've heard that sometimes the military can get it paid for. I've heard Kaiser's had some success depending on the Kaiser and the clinician. Outside of that, basically, I'm hearing that no insurance is covering it still. There is an ICD-10 for HAT, but currently there's no CPT code. And so that's been the hang up right now. I actually talked with an insurance type guy about how it is you go about getting CPT codes, and it actually sounds like it's not that complicated, but it just hasn't been pursued / accomplished yet.
Great. And then a question from Don Rubin: mutations in, I think it's non-coding sequences, may pose additional challenges in designing therapies. Any potential insights into designing such therapies?
Well, I guess what I would say is, at least in my impression of non-coding variants, certainly there's in GWAS where there's, impactful non-coding variants that, it's hard to link to genes and then you're sort of stuck. Generally speaking, when we're doing this, the non-coding variants we identify tend to impact splicing or gene expression, or and so there's still functional impacts that can be targeted. I don't know if this is related to the non-coding sequences that distinguish duplicated alpha from wild type alpha. In those cases, what we're really identifying is people who have even higher alpha tryptase expression relative to modest alpha tryptase expression, because those non-coding variants tag that promoter that's overactive and driving overexpression of the replicated alpha tryptase gene. But certainly, I agree, like among those identified, particularly like in GWAS, where there's even a strong linkage with a non-coding variant that's in some region of the genome, it's hard to know which of the genes it's actually linked to. And sometimes it's not even linked to a gene in the region, but it's one that's far off in the distance, and it takes years to sort that out.
Great. Well, I think we're at the end of our time. John, I think that if anybody else had questions, they can go through me to get to you, or it's pretty easy to find your contact info as well. I'm sure you get lots of emails about HAT and potential patient mechanistic issues related to their genetics, et cetera. But really appreciate your time. You're also an awesome example of, how glamorous, I don't know about wealthy, but at least glamorous you can become if you graduate from internal medicine at UC San Diego.
UCSD, yeah.
And it is wonderful to see how you've done everything since. So appreciate it. And thank you everybody for joining today.
Thanks again for having me.
Thank you so much.
Thanks.